This will take a moment to load here. There we go. Okay, I am ready. Let me see if I can share screen.
Yeah.
Can you see that?
Yeah.
Okay,
Your screen
wonderful.
is in front of us. Yeah. We can see the title.
Good.
Okay. So let me introduce you. So it's a real pleasure to have Professor Dr. Hiten Madhani with us. He is originally from UK. He was born in London, UK, grew up in Seattle, and attended Stanford University in US. He received his MD and PhD degrees from UCSF. And after postdoctoral work at MIT Whitehead Institute, he returned to UCSF as a faculty in 1999. He is currently a professor in the Department of Biochemistry and Biophysics. He was recently elected as Fellow of American Association of the Advancement of Sciences, and he is also Fellow of American Academy of Microbiology. Thank you very much, Hiten, for joining us. It's a real pleasure to have you. Over to you.
Pleasure to be here. Apologies for my confusion.
No problem.
Okay, what's shown in the first slide here is one of my favorite places to go hiking, which is called Muir Woods, where the tallest or largest organisms on the planet live, which are these coastal redwoods, and it's a favorite hiking place of mine. See if I can get my slides to work good. So geneticists have long been fascinated by epialleles, which are Mendelian traits in which the two alleles have identical DNA sequences. Shown here are flowers of the yellow toadflax, Linaria vulgaris. And at the top in panel C here, you can see what a wild type flower looks like, which has bilateral symmetry. But below it, you can see a flower that looks quite different, that has radial symmetry. And these two flowers were initially classified as different species by Carl Linnaeus himself. In fact, they're the same species. One is a mutant, but not a classic DNA mutant, but an epiallele caused by DNA methylation and silencing of a gene that mediates the switch from a radial symmetry in flowering to bilateral symmetry. So these epialleles that are found often in plants have limited genetic stability. In other words, over several generations, often the epiallele is lost and the phenotype reverts back to wild type. So this raises the general question of whether an epigenetic mark can mediate the evolution of traits. For this to happen, of course, you have to have inheritance of information, random variation in that information, and then natural Darwinian selection to select out the winners in the evolutionary game. So what I'm going to discuss with you today is as follows. This is the outline. I'm going to talk about, give you some background on DNA methylation in mammals, introduce a model system, which is a yeast called Cryptococcus neoformans, tell you about the basic features of its DNA methylation system, and then a paradox followed by an evolutionary surprise, leading to a hypothesis, and then data supporting that hypothesis. And then if there's time at the end, I'll describe some recent structural insights. So when I talk about DNA methylation, in this case, I'm talking about 5-methylcytosine. So this is cytosine with a methyl group at the 5 position. And what you should know is that 70% of the cytosines in the human genome are actually methylcytosines and not normal cytosines. So it's the majority of C bases in the human genome. It was proposed in the 1970s that methylation, DNA methylation of cytosines might enable the memory of prior developmental events. And in fact, it might even explain developmental memory. So the hypothesis put forth by Holliday and Pugh and also Art Riggs was that there would be two types of enzymes that do DNA methylation in eukaryotes. One that they called de novo, which would be able to modify unmodified DNA. So regular old DNA could be modified by methylation, perhaps on both strands, but then due to DNA replication, it would be diluted out once the de novo enzyme had faded from the scene. They further proposed that this initial activity of the de novo enzyme could be then essentially propagated epigenetically in a form that involves memory by a second type of enzyme that could only recognize the hemimethylated DNA produced by DNA methylation. And it would somehow recognize that there was methylation on one strand and not the other, and then place methylation on the other strand to restore the original state. And so this was called a maintenance enzyme. So it turns out this is not how developmental biology works per se, but nonetheless, DNA methylation has very many important roles in, for example, mammalian biology, plant biology, and so on. And part of it is to silence repetitive DNA. So about half the human genome is derived from transposable elements and repeats. And these have to be silenced because they're deleterious. And they're silenced by DNA methylation. DNA methylation also plays an important role in chromosome segregation and centromere function, which is not well understood, as well as monoallelic gene expression. So for example, in human females, one copy of the X chromosome is silenced in a process called X inactivation, and DNA methylation plays a role in maintaining this. So despite the fact that DNA methylation doesn't really explain developmental memory, this two enzyme paradigm in vertebrates has held up. So the de novo enzyme exists, and it's called DNMT3, and the maintenance enzyme is called DNMT1. DNMT3 is actually more complicated than just a single protein. It's actually a heterotetramer of either DNMT3A or 3B with a pseudoenzyme, a catalytically inactive enzyme called DNMT3L. DNMT3A mutations often drive acute myeloid leukemia. So DNMT3A and 3B are targeted by several mechanisms. One is that DNMT3A, 3B heterotetramers recognize a histone modification, or rather the lack of a histone modification. On histone H3 lysine 4, if lysine 4 is unmethylated, it forms a binding site for the ADD domains of these enzymes, which actually activates their catalytic activity. Small RNAs called piRNAs are also involved in establishing DNA methylation in the male germline of mammals through a process that at the mechanistic level is not well understood. The maintenance enzyme in mammals is called DNMT1. It's this large protein. It's been crystallized and characterized. And again, it's a large, complicated protein that has a catalytic domain and a number of other domains. And even this enzyme is more complicated because DNMT1 works with a cofactor called UHRF1. And this cofactor has a number of protein domains, again, one of which recognizes hemimethylated DNA, and another, a tandem tutor domain that recognizes another histone modification called histone H3 lysine 9 methylation. And this protein is overexpressed in tumors and displays oncogenic driver activity. So, so here's a little cartoon of maintenance methylation in which DNMT1 recognizes hemimethylated DNA and methylates it. UHRF1 also recognizes hemimethylated DNA and helps recruit DNMT1 through a direct interaction. And also UHRF1, which is not shown here, has an activity called a histone H3 ubiquitin ligase activity, which is then ubiquitination of histones is also recognized by DNMT1 and functions as a further recruitment mechanism. So most of our understanding of this process of cytosine DNA methylation comes from studies of just a handful of species. And this is mammals, as I described to you earlier. Much work has been done in plants, particularly Arabidopsis, which has a fascinating DNA methylation system that's driven by RNAi. As well as in the fungal kingdom, there's foundational work from Eric Selker at the University of Oregon showing that in the bread mold Neurospora crassa, DNA methylation there is directed by histone H3 lysine 9 methylation. Those are some of the first studies to show a connection between a chromatin modification, a histone modification that is, and DNA methylation. So that's all the background I'm going to give you about DNA methylation, but myself being a simple-minded yeast geneticist would like a yeast system for studying these silencing mechanisms because of the experimental tractability of the system. And so Saccharomyces cerevisiae, the dominant model unicellular eukaryote, unfortunately lacks DNA methylation and a number of other gene silencing mechanisms. Fission yeast, Schizosaccharomyces pombe, has many of these modifications and RNAi, but it lacks DNA methylation. The yeast I'm going to tell you about, which is called Cryptococcus neoformans, in contrast, is more complex and harbors all of these chromatin modification systems as well as DNA methylation. So Cryptococcus is a yeast, a budding yeast, but it's very it diverged from the model yeast, Saccharomyces cerevisiae and Schizosaccharomyces pombe, by about 600 million years. So Saccharomyces and pombe are ascomycota, which includes most fungi that you've ever heard of, whereas Cryptococcus is part of this other subphylum in fungi called the agaric basidiomycota, and within that, this group called the agaricomycotina, and that includes all mushrooms. So Cryptococcus is far more closely related to a mushroom that you might eat than any yeast that you've heard of. The reason why people are interested or have been historically interested in Cryptococcus is that it's an important human pathogen. It's the most common cause of fungal meningitis in humans, and it's an opportunistic pathogen, meaning that individuals who have an intact immune system are not susceptible to infection. However, individuals with a weakened immune system are. And this includes people who lack T cells. So for example, patients with end-stage AIDS. And this organism is inhaled and you and I clear it thanks to our adaptive immune system, but without CD4 T cells, the organism can't be controlled and spreads into the blood, into other tissues, and in particular the brain, to cause a meningoencephalitis that is uniformly lethal if not treated, and displays fairly high mortality even with optimal treatments. So as a result of this, Cryptococcus causes about 15% of HIV deaths annually, about 181,000 deaths per year, spread worldwide, but of course concentrated in countries that have the highest rate of HIV positivity, and also the weakest systems in place for treatment. Okay, so I'm going to talk about not Cryptococcus as a pathogen, but as a basic science platform. And over the years, it's allowed us to investigate many questions based on the fact that it has features of its biology that are conserved with other organisms, but were lost during the evolution of Saccharomyces cerevisiae. And today, again, the focus is going to be on DNA methylation. So turns out Cryptococcus, its genome encodes a single DNA methyltransferase, or DNMT, which is called DNMT5. And here's the protein. It has a catalytic domain, a ATPase domain that's related to those of chromatin remodeling enzymes, switch-SNF ATPases, and also a reader domain that recognizes chromatin marks. So this enzyme, when you knock it out, results in the loss of all DNA methylation in Cryptococcus. So in Cryptococcus, this is a browser shot of a chromosome, chromosome 10, and what's on the y-axis is the fraction of every CG on this chromosome that's methylated. And what you can see is that there's a cluster of methylation sites in this portion of the chromosome, which is rich in repetitive elements. And this cluster corresponds to the centromere. And every chromosome has a region like this, which corresponds to the centromere, and it's heavily methylated by cytosine methylation. There's also a little bit of methylation in subtelomeric regions, which are at the end of the chromosome. And then if you delete this, the enzyme, the gene encoding this enzyme, you lose all of it. So the methylation is restricted to these regions and plays a role, it turns out, in gene silencing. And probably the best evidence that that's really the primary function of DNA methylation in this yeast is that Cryptococcus neoformans has a number of active transposable elements, full length and of course remnants at its centromeres. And it has both DNA methylation, 5-methylcytosine, and RNAi as two parallel systems that silence these deleterious genomic elements. A sister species called Cryptococcus deuterogadii has lost all of its active transposons. And so what's happened here is that the transposons have been degraded. They no longer are intact. They've acquired mutations so that no transposons appear to be active in this species any longer. And correlated with this is a complete loss of 5-methylcytosine and RNAi. So the gene silencing systems are dispensable once the transposons have been lost. And we think this is simply a chance event that happened in this particular lineage. So the first question we asked is how 5-methylcytosine is targeted to the right places. That's sort of a standard question to ask. And chromatin biology is how different portions of chromosomes acquire their specializations. And I won't show you all the data, but this is an example of some browser tracks showing where particular chromatin modifications are in Cryptococcus. And as you can see, that at the centromere, where we see most of the DNA methylation, it's highly enriched in this highly conserved marker of centromeres called, again, histone H3 lysine 9 methylation. And then when you knock out this enzyme that's called CLAR4 in Pombe, and we gave it the same name in Cryptococcus, you could see that you lose all the H3K9 methylation. And this was a good candidate for something that might recruit DNA methylation, we thought, because if you look at the enzyme DNMT5, it has a catalytic domain, and it has just upstream of it this chromodomain, which is a domain that's a common reader of methylated lysines on histone tails. And then we thought perhaps this swish-snif ATPase domain would be a chromatin remodeling domain that would help move nucleosomes out of the way so the enzyme could modify DNA. That, by the way, turns out to be completely wrong. Again, I'm not going to show you the data, but as a result of a number of biochemical and genetic experiments, we came up with the following model, which is that this enzyme, DNMT5, is indeed recruited by histone H3 lysine 9 methylation to its sites of action. But it's recruited by two different mechanisms. One is a direct mechanism where its chromodomain of DNMT5, that's what CD stands for, directly binds this methyl histone mark. And an indirect pathway where a protein called HP1 or SY6 in Pombe also recognizes this histone mark and then in turn binds DNMT5. We also found an ortholog of UHRF1, that human protein that I told you was involved in maintenance methylation, that in Cryptococcus recognizes hemimethylated DNA and promotes DNA methylation. And so this is basically setting the stage of this genetic pathway and a biochemical pathway for how DNMT5 is recruited to the right places at the right time. Okay. So although I'm not going to show you any data, I'm going to show you an assay that we like to use, which is an old-fashioned southern blotting assay. So what's shown on the right here are a couple southern blots. And lanes in the southern blot are wild type, a knockout of the enzyme DMT5, and a knockout of the H3K9 enzyme CLAR4. And so we used a restriction enzyme that has CG in its recognition site, but where cutting is blocked by methylation of that CG. And so if you cut with that restriction enzyme in wild type cells and you probe with a particular unique probe at one of the chromosomes, what you get is a band that's 3.5 kilobases in size on a southern blot, as shown over here to the right. So U at the bottom here refers to a unique probe. And then, and that's because there are two unmethylated sites here and here, and in the middle is where the probe sits. If you remove all methylation by knocking out the methyltransferase, now you can see that there's a much smaller restriction fragment that the probe sits on. That's this 1.5 kb band you see at the bottom here with the unique probe, probe U. Above is a probe R, stands for repetitive probe. So this is a piece of a transposon, basically, that recognizes fragments, restriction fragments on many different chromosomes to get many different bands. Nonetheless, if you get rid of DNA methylation, what you can see is that the bands collapse to a lower molecular weight pattern, because now you're cutting sites that are closer to the probe. And as you can see, if you get rid of H3 lysine 9 methylation, you reduce but don't eliminate DNA methylation. Okay, so the first question we asked is whether DNMT5 is a maintenance enzyme, a de novo enzyme, or both. And what I should mention is that DNMT5 is the only cytosine DNA methyltransferase encoded in the genome. So we assume that it would either be a de novo enzyme or both a de novo enzyme and a maintenance enzyme, right? So, and I should mention that in Neurospora, which has been studied by Eric Selker, as I mentioned earlier, it appears to be a de novo enzyme and perhaps also has some maintenance activity. Okay, so here's an experiment we did. We took wild type cells and we knocked upstream of the gene, the DNMT5 gene, a promoter, a galactose regulatable promoter. And we did it under conditions in which the promoter was off. So you knock this promoter into the genome, and you grow up the transformants that turn out to be the knockout, and we call those repressed, for R for repressed. And then, and of course, there's no DNA methylation here because the methyltransferase is off. Then we turn on DNMT5, the gene, and we wait either 20 or 45 generations, and we ask whether methylation comes back. And so what you can see on the southern blot on the right, the first lane is wild type. The second lane is the knockout, where you have no methylation. And R, the lane with R, is the repressed lane, where you can see, again, no methylation as expected. And of course, no protein. This protein has a tag on it, a flag tag. We now induce expression of the protein for either 20 or 45 generations. You can see that no methylation returns at all. So you might say, well, perhaps this is because you tagged it and it's overexpressed, perhaps it's a non-functional protein and you just didn't do the experiment right. And so we did the experiment a different way, which was to start with the gene on. So it's a galactose-driven DNMT5, which is induced. So I0 here means induced. And what you can see in that lane, it looks just like wild type. So this is a perfectly functional allele of this protein, plenty of expression, and methylation is normal, as in wild type. Then we turn it off and wait until DNA methylation has been diluted out for many generations. So that's R for repressed. And then we turn it back on again now for 40 or 90 generations, so even more generations. And what you can see is that once again, DNA methylation doesn't come back, either at the unique probe or with the repetitive probe. So once you've lost DNA methylation, it doesn't seem to come back in this organism, even when you reintroduce the enzyme. We did the experiment a different way. Rather than having an overexpressed allele, what we did is we knocked out the catalytic domain of DNMT5, and then we reintroduced, that's what RI stands for here, the catalytic domain along with the promoter. And again, if you look at the southern blot on the right, the first two lanes are the control, wild type, and the knockout. And then the reintroduced DNMT5, you can see, even though it's expressed, we tagged it again, it does not restore DNA methylation consistent with the results I described for you before. So once you've lost DNA methylation, once again, it does not return if you bring the protein back into the cell. I've been showing you old-fashioned southern blots, but we can use any number of techniques to examine DNA methylation. Here we're using what's called whole genome bisulfite sequencing. So it's a genome-wide method, and what you can see again is wild type has a lot of DNA methylation at the centromere. The knockout loses it, and when we reintroduce DNMT5, you can see that DNA methylation doesn't return to the centromere. If we look genome-wide at all the methylation sites we can assay with this whole genome assay, what we can see is that the knockout loses DNA methylation, but if we look at two different isolates of the reintroduced strain, you can actually see 2 out of several thousand cytosine becoming remethylated. So it means that a tiny bit of de novo methylation can actually occur in this strain. We also did atomal sensitivity mass spectrometry with Petra Hodgkova in London. And again, we could see that there was no detectable cytosine methylation when we knock out DNMT5, When we reintroduced it, although it couldn't be quantified because it was below the quantification standards of the experiment, we saw a little tiny peak, which we think is consistent with this little, very small amount of DNA methylation when we reintroduced DNMT5. And I'll come back to that later. Okay, so this reintroduced enzyme is functional. For example, we can see that it still localizes to centromeres. So this is a whole genome chromatin immunoprecipitation where we localize the protein to chromatin. Here's the location in wild type. This is the centromere of chromosome 13. When we reintroduce the protein, you can see that we can still chip it quite well to the centromere. So it binds chromatin, it's just not able to trigger globally methylation. We can even do a genetic experiment to show that it's a functional enzyme. So if we take the reintroduced DNMT5 strain and we cross it to a wild type strain where we've marked the centromere, and we take progeny that have this wild type centromere and this reintroduced enzyme rather than the wild type allele. When we look at the progeny by southern blot, you can see that the progeny all have the band of the methylated size as opposed to the unmethylated size. So the reintroduced allele is functional when you bring it together with a methylated centromere. We can also do a cute experiment where we can ask whether passage through meiosis is able to reestablish methylation. Often DNA methylation is established pre-meiotically during development, whether you're talking about plants or animals. And so I'm just going to go over the panel on the right here. And all we've done on the right here is started with a marked centromere that's in the knockout strain, okay? And so this centromere has no DNA methylation because it's in a knockout strain. And we can ask the question, what happens when we go through meiosis and bring this centromere together with wild type DNMT5 and look at those progeny? And as shown on the southern blot with this unique probe, you can see that 3 different progeny that we assayed where we have a wild type enzyme together with a centromere that is unmethylated, you can see that the centromere remains unmethylated despite the fact that the cells have undergone this developmental pathway of meiosis and sporulation. So in any number of ways, we found that once you lose DNA methylation, it doesn't come back, which all suggests that DNMT5 is a maintenance methylase, a rather pure maintenance methylase that once you've lost DNA methylation, that's it. So we asked whether it really was a maintenance enzyme by expressing it in yeast, Saccharomyces cerevisiae, which of course doesn't have DNA methylation, and purifying it. So it's a difficult enzyme to express in large amounts. So we were able to do it in yeast initially. And then we purified it and mixed it together with substrates. Oligonucleotides that contain 3 CG dinucleotides that were either unmethylated, hemimethylated on the Watson strand, hemimethylated on the Crick strand, or fully methylated. And then we simply mixed the enzyme with the DNA and buffer and S-adenosyl methionine, the universal methyl donor in cells. And we found that nothing happened, no incorporation of radioactive SAM into DNA. But when we added ATP to the reactions, we saw beautiful methylation. And the methylation only happened on hemimethylated substrates. So in vitro, this enzyme is a pure maintenance enzyme that unusually is ATP dependent and also SAM dependent. And it, again, simply will methylate hemimethylated CGs with reasonable efficiency and kinetics. In fact, we can give it an unmethylated substrate and wait as long as we wish. And as you can see on this log scale, we don't get any incorporation above background. And so this assay has about a 3 log sensitivity. So we think the enzyme is, roughly speaking, at least 1,000 times, if not more, specific for hemimethylated DNA than unmethylated DNA. And this is considerably more selective than other maintenance methylases that have been described in the literature. Okay, so the big question now, which you should be asking yourself is, well, how in the world could methylation ever be established in a species with a maintenance-only paradigm? This doesn't make any sense, right? And so we explored this in a variety of ways, and we'll start with the phylogenetic approach, which gave us our first clue. So what I'm going to show you is a phylogenetic tree, and what you can see is that species that are close, all the human pathogens have just DNMT5. I told you about Cryptococcus deuterogadii. It also has, which has lost cytosine methylation and DNMT5. A non-pathogenic species, Cryptococcus amylolentus, and another one called Cryptococcus wingfieldii, just has DNMT5, and there's yet another species that seems to have lost DNMT5, and we predict will have lost DNA methylation. But if one looks further out on the phylogenetic tree, what you can see is a whole number of species that have DNMT5 and also a second predicted cytosine methyltransferase that we call DNMTX. And what you can see from looking at this phylogenetic tree is that the DNMTX gene was lost on this branch of evolution. So the ancestor clearly had this maintenance enzyme, DNMT5, and a second enzyme. So the obvious hypothesis was that perhaps DNMTX is a de novo enzyme that existed in an ancestor, but the gene was lost. And so to test this, we introduced DNMTX genes from existing yeast into that reintroduced Cryptococcus DNMT5 strain. So this strain, again, can maintain DNA methylation, but it can't generate it de novo, at least not efficiently. And so we cloned and overexpressed DNMTX from 3 different species into this strain, and we asked whether we saw de novo DNA methylation. And so the results of this are shown here. This are data for centromere 9. The parental strain is here, and we use 2 different methods to look at DNA methylation. And you can see that there's no methylation at the centromere in the parental reintroduced strain. But if you introduce DNMTX from Cryptococcus pinus, Quaniella mangrovensis, or Cryptococcus bestiola, you now see methylation de novo at this centromere and many other centromeres and even some subtelomeric regions. So clearly, DNMTX, when introduced into Cryptococcus neoformans, can trigger DNA methylation de novo, which then, of course, can be maintained by this maintenance enzyme, DNMT5. And so that's our model for how methylation started. And so the next question we asked was, well, how long ago did this happen? And if you take estimates for when these branches, these are just published elements for when these branches occurred, which is, of course, just an approximation, it seems that DNMTX was lost between 50 and 150 million years ago. What's shown here is a depiction of how many genera there are in thousands during our current period, the Phanerozoic. And the upside down yellow triangles represent mass extinctions. And so DNMTX was lost sometime between 50 and 150 million years ago. And so what that means is that that gene was lost probably when dinosaurs roamed the earth. And so the non-avian dinosaurs didn't make it to the present, but Cryptococcus has, as did our ancestors. And it did it since the Cretaceous, again, without the benefit of a dedicated de novo enzyme. So this raised yet another question, which is, well, how is it possible that 5-methylcytosine has been propagated after the ancient lost millions of years ago of DNMTX? It seemed quite unlikely that there would be an initial cytosine methylation pattern that was set up that was perfectly propagated for many, many generations, right? So we did 2 types of experiments to address this question. One was a laboratory evolution experiment, and the other was a phylogenetic analysis. So the laboratory evolution experiment was very simple. We start with wild-type Cryptococcus, and we simply grow it. So we grew it for 120 generations, and then without bottlenecking, and then took individual colonies, so the progeny of individuals, grew them up just enough so that we could do whole genome bisulfite sequencing. And we did this in replicate, and we did this extremely carefully. So we could really measure quite accurately whether methylation had been lost or even gained during laboratory propagation, and whether it was random. And so here's an example of part of a centromere, and the vertical bars show you where cytosine methylation is in this region. And what you can see is an example of where in the parent there was DNA methylation. In one of the colonies, it was maintained, but in another colony, this particular site was lost. Even more rarely, we saw examples where in the parent, DNA methylation was clearly absent, and we actually look at the sequencing reads directly to make sure that this is the case. And then we found an example of a colony where there was de novo gain of methylation. And so the key is to quantify all this and calculate rates. And so what you can see is that the overall picture is that there's a high stability in the laboratory of methylation with losses occurring much more frequently than gains. So out of over 4,500 cytosine methylations that we could assay in the parent, 99% of them are intact after 120 generations. For one colony, there was 39 loss events, and in the other 50 loss events. And then there were much rarer gain events, 5 in one case and 5 in the other. And these all are random. There's no significant, overlap between these events in the two different colonies. So if you do the calculation, it turns out that the rate of loss is about 20-fold higher than the rate of gain. And so that shows that the system is not in equilibrium. It's like a chemical reaction that's not in equilibrium. And so to maintain the situation, natural selection must be required to maintain the steady state level. So that's the first piece of evidence that DNA methylation in Cryptococcus is under natural selection. And this is just to show that there's no significant overlaps between the two colonies. The two that we see between the two colonies are what you'd expect. So this really appears to be something like random Darwinian evolution, or variation rather. So we also investigated the natural evolution of 5-methylcytosine. So for this, we took a group of isolates of Cryptococcus, which has been heavily studied in terms of its population genetics. We took isolates from 4 different clades that had a common ancestor about 5 million years ago. And we assembled their genomes by long read sequencing, and we measured DNA methylation in these isolates. And we asked 2 questions. One is whether methylation is conserved across evolutionary timescales. And the other question we asked was whether it was subject to any natural selection based on where the methylation was conserved. I'm not going to go through the gymnastics required to do this study, but what I will say is that it was a lot of work and involved collaboration from multiple groups. So if I can go back a slide. Oops. So if I can get the slide to go back, let's see if this might take a moment. That's the large. Yeah. Okay, so what's shown here in this Surcos plot is an analysis where we take centromeres. So the outer circle describes, in this case, centromere 4 from all of these strains. The inner circle here describes the regions where we can measure cytosine methylation. And then color-coded are the lines that show whether methylation is conserved. So we have to find regions that can be aligned between centromeres and look at conservation of cytosine methylation. The reason why we have to do this analysis this particular way is that centromeres, because they're repetitive, evolve quickly, and they're impossible to align end-to-end across these different species or isolates. And so we were left with doing local alignment of both DNA and DNA methylation patterns. We could then ask for a given centromeric, whether this sharing of methylation between isolates was more than you'd expect by chance. And so the complication here is that a given centromere, because of their rapid evolution, might be conserved only between 2 isolates, 3, 4, and so on. Very few alignments are found in all of these strains that diverged 5 million years ago. But nonetheless, you could ask, for example, in instances where a sequence can be aligned in 6 different centromeres, what fraction are methylated, and how much higher is this than what you'd expect by chance? And so that's what the expected and observed indicate here. And you can see for every class of alignment that there's much more conservation or sharing of methylation patterns than you'd expect by chance, which supports the notion that there's natural selection operating on these methylation patterns. We also asked whether there was more methylation on full-length retrotransposons. After all, if it's important to silence transposons, it should be the full-length transposons and not the defective mutant transposons that need to be silenced. And so what's shown here is the fraction of CGs that are methylated in full-length centromeric transposons versus other centromeric sequences that are not transposons. And what you can see is that for every strain, there's significantly more methylation density on full-length transposons rather than transposon remnants, which is, again, consistent with the hypothesis that natural selection is operating to maintain DNA methylation. And so what we have is a situation where you have something equivalent to Darwinian evolution, but it's not DNA sequences being simply acted upon by evolution, but a DNA modification. So in this sense, you can think of 5-methylcytosine as a fifth base of DNA that's under evolutionary pressure or evolution. So I've shown you that there's inheritance mediated by this enzyme. There's natural variation in the form of loss and gain, and then there's natural selection that operates on this protein on regions of the genome that are particularly important to silence, namely transposable elements. And here's some references on our recent work on the enzyme that's all published. But I'll share with you now is some unpublished data on how DNMT5 as an enzyme actually works. And the questions are, what explains its substrate specificity? It's extremely specific for hemimethylated DNA. How is this substrate recognition coupled to its ATPase activity? And how is this ATPase activity then coupled to the DNMT activity? We don't have all the answers, but we have some hints. And the hints have come from solving with Juncheng Wang and Dinshaw Patel at Sloan Kettering Cancer Center cryo-electron microscopy structures of DNMT5, either in an Apo form, in a binary complex with hemimethylated DNA, and then in a binary complex with hemimethylated DNA along with an ATP and SAM analogs. And what I'm going to show you now is just a couple quick movies that describe the differences that we see between the structures and a model for how we think this all works. So this is the Apo form of the enzyme, and what's in the middle is, and in green is the DNMT catalytic domain, and abutted to it is this target TRD or target recognition domain. They're held on a sort of platter formed by this ring domain that seems to form sort of a structural scaffold on the back. The switch-SNF ATPase is the rest of the structure, believe it or not. And there are these two Rec-A domains here that are conserved in all superfamily 2 helicases and switch-SNF ATPases that when they bind nucleotide, as you'll see, come together and close. And that those conformational changes are coupled to changes in inserts in this in these two Rec-A domains. And one insert is this HD or helical domain, and the other insert, it's actually 2 inserts, involve both an alpha beta domain and a helical domain that couples motions in the Rec-A domains to changes in the conformation of the DNMT domain. And I'm hoping that this movie will play. This somewhat slow computer I have here. I have the spinning rainbow ball on my screen, so we'll see if it loads in a moment. And there's actually 2 2-minute movies I'd like to show you along these lines. Come on, movie. Seems to be not cooperating at the moment. I can't even escape out of it. Never a good sign.
So in the meantime, may I ask you a question while the
please.
Yeah. So this DNMT5 mutant,
Yes.
is it viable? Do you see any phenotype like chromosomal instability or transposons getting activated, etc.?
Yes. Good question. Something happened here on my screen. Okay, so let me quit out of it. Let's see if it'll play. The answer is we don't know. We have seen some increased sensitivity to chromosome destabilizing agents, which we infer to mean that there is a chromosome instability defect, but we haven't done the assays to look directly. And we have not also done the assays to look directly at transposon mobilization. That requires some engineering that we can actually do much more easily now because we have CRISPR working extremely well in this organism. So to be determined, but those would be good experiments. Let's see if it plays now. Okay, can you see that?
Yeah.
Okay. So the enzyme is a giant donut, as you can see, and again, has the domains I mentioned. And so now we're going to morph to the binary complex that's bound to a DNA oligonucleotide. And what you can see is that there's a residue here, asparagine 669, that forms a steric clash with the methylated base of the parental cytosine. And so in the binary complex, we see that that residue moves out of the way, and in a fashion that seems to be concerted or triggered by that, although we don't know, there's a restructuring of a hydrogen bonding network in this part of the enzyme. And so the DNA appears to be stabilized by specific hydrogen bonding interactions between the methylated cytosine base and the G just above it, as well as some interactions with the DNA backbone. The cytosine to be methylated becomes partially flipped out of the double helix, as you can see here, not completely, but partially. And that partial flip out, which has never been seen before in a cytosine methyltransferase, appears to be stabilized by another hydrogen bond. And again, this is just showing you some of the hydrogen bonding network that seems to interact with the DNA. Associated with the binding of DNA are other conformational changes in the target recognition domain. And one of these involves the movement of a loop that we call the allosteric loop, which then forms some hydrogen bonds with a conformation of the switch SNF ATPase, which is shown over here to the left. And so what you're going to see is interactions between the switch SNF ATPase and the target recognition domain, and even the DNA, that only happen once the DNA is bound. And the function of those is to stabilize a rotated form. So in the unmethylated state, without DNA, the enzyme has one conformation, but with DNA, this switch SNF ATPase, this half of it, does a rigid body rotation of, I don't know, about 20, 25 degrees. And so it's a very striking and simple conformational change that occurs when the DNA binds. And that conformational change changes the relative orientation of these two Rec-A-like domains. And we think that allows the enzyme to now actually bind nucleotide. And so now we're going to, if I can get this one to load, we're going to morph from that binary complex to a complex that has a nucleotide and an analog of S-adenosyl methionine bound to the enzyme. So you can see what happens when nucleotide hits the enzyme. I'm going to try, okay, this is dangerous, but I'm going to try it. I'm going to hit play. Dare I? Oh, I have the spinning ball again. Well, we know it will work. It'll just take a couple minutes. But what we're, oops, maybe that was a bad idea. Backing up. Okay, here we go. I hope. Yeah, so there's nucleotide analog binding, and you'll see what happens is that the two Recce domains close when nucleotide binds. And this is a very standard, ATP binding site with your standard Walker A and Walker B motifs, and you'll see that all of those motifs are present, and all the hydrogen bonds you would expect from side chains in those motifs are there. So interactions with the base, the standard interactions holding magnesium and the phosphates, and also interactions from the other Recce domain, the Recce2 domain. What's interesting is then what happens when these two domains close. And you can see a number of concerted motions. This is from the front. We can turn the enzyme 90 degrees. And yes, now we can see acetinosyl homocysteine, which is a SAM analog, is actually the product of the reaction. They're being used as an inhibitor. And you can see that the DNMT domain moves up relative to the DNA. And it's almost like a mechanical device in action. It's a real example of allostery. And so when we see this domain move up, you can see that there's a loop that now becomes visible in the structure, and that loop penetrates into the major groove of DNA, seemingly poking that partially flipped out cytosine all the way out of the double helix. And then there's a number of hydrogen bonding interactions that stabilize this invasion of the double helix by the protein again, and then there's maintenance of this flipped out state by specific hydrogen bonds that we can infer. And so there you can see the analog acetinosyl homocysteine. And the other thing that becomes apparent in the structure is a cysteine, which is found in the active site of all cytosine methyltransferases. And this residue actually in the reaction mechanisms forms a covalent intermediate with the base, allowing methyl transfer through the substrate. So we're now doing a number of biochemical experiments on purified enzymes to test some of the ideas that I've put forth here. In particular, we would be very excited to see if that mutating that asparagine allows us to get some de novo activity from this enzyme. We think that at least the exquisite specificity is partially explained by the target recognition domain reconfiguring in response to a clash produced by the methylated parental cytosine and asparagine 669. And this reconfiguration seems to promote a rigid body rotation of the second Recce2 domains that allows the two Recce domains to be juxtaposed in such a way that nucleotide can be bound. And we suspect that the closing of the ATPase appears to trigger a change in the DNMT domain that puts it in a precatalytic conformation. So these are all just ideas from a structure. Structures, in my view, generate hypotheses. And so there's much biochemistry to be done to test these ideas. So that's it. All of the work I showed you that was done in vivo in cells was done by a very talented postdoc, Sandra Catania, originally from Sicily via the UK. Philip Domestic was a student who did all the biochemical experiments and more that I described to you. Christina Cuomo at Broad Institute and her team did the long read sequencing. Jonathan Pritchard and his postdoc Harold Pimentel is now a professor at UCLA, did the analysis of the phylogenetics. Petra Hodgkova did the mass spec on DNA methylation. Caitlin Stoddard in Geeta Narliker's lab, one of my colleagues in the department, helped with the DNA methylation assays. And John Yates is our long-term mass spec collaborator, and Dinshaw Patel is our structural biology collaborator on this project. These are our funding sources. This is a picture of the lab before the pandemic, as you can see by all the smiles. It was a Halloween party that people were putting on. So anyways, I will apologize again for being late, and be happy to answer any questions people have. Let me
Thank you very much.
About my share here.
It was a wonderful talk. So questions. I see
Yeah.
Dr. Afzal has a question.
Yeah, thank you very much, Professor Hiten. It was really so insightful talk. Learned many things, very basic about the DNA methylation. Now, I was wondering about forgive me for my ignorance about the basic yeast biology.
Mhm.
I mean, you showed that very related yeast species. For example, we know that Cerevisiae don't contain any DNA methylation, but this one contains. So first I was wondering, there are not transposable element in Cerevisiae genome,
There
or they are
are.
not?
There are, yes. In fact, the
So-
first described retrotransposon was demonstrated to be such in Cerevisiae, Jerry Fink's lab.
So how they are silenced?
Yeah, well, nobody knows. I mean, there are no dedicated silencing systems in Cerevisiae for transposable elements. There are many mutants that people have isolated that cause them to transpose at higher frequency. So the hypothesis is that if you think about it, the transposable element doesn't want to kill its host, otherwise it will not survive. And so it could be that there's sort of an evolutionary détente there, where despite the fact that Cerevisiae lacks DNA methylation or silencing systems, the transposons have reduced their activity so as to not be quite so toxic. The other thing that I think to keep in mind is that the way transposons spread through populations is certainly in fungi, is through outcrossing, through sexual reproduction. And so most fungi actually grow, for most of the time, they grow clonally, right? The outcrossing is important for getting rid of deleterious alleles and reshuffling the genome, but it comes as a danger, right? There's a danger of catching a sexually transmitted disease, which is a transposon, right? And so what's interesting to me about Cerevisiae is that it has something called mating type switching, where a single clonal population can go through sexual reproduction by changing its mating type and mating with its progeny, essentially. So I've often wondered whether organisms that don't silence transposons by these mechanisms have found some other way of preventing spread through their population.
Another thing I was thinking, it was really, clever choice to choose this DMT5, in its very ancient, evolutionary place. I mean, I always thought these DMT class, they are de novo methylases, but your data clearly shows in the simplest form, they are rather maintenance. And you also showed the other enzyme related, which was DMTX. So that was rather de novo. So how closely related they are? They are derived from the same gene, or they are different?
They're completely unrelated. I mean, they're as far as you could be on the evolutionary tree of DNMTs. So DNMT5 is actually more closely related. It's out there on its own, actually. And then DNMTX is more closely related, I think, to DNMT1, if I remember correctly. So it's all shuffled around.
All right, thank you so much.
Other questions?
I have a question.
Please.
Go ahead.
So, since you mentioned that DNMT5 was the only one copy gene present in yeast for maintenance methyltransferase activity, I was wondering how come that it was lost about 150 million years ago in the evolutionary tree, and since we know that DNA methylation is important for gene regulation, how these strains survive through all these years?
Right. Well, so one.
The DNMT5.
Let me just say that we don't have any evidence in Cryptococcus that it's involved in gene regulation per se. We really think that the only function is in suppressing transposable elements. So you could really ask the question a different way, which is to say, well, why do you even have de novo enzymes? And presumably you have de novo enzymes because at some point there's a need to recognize new transposons in the genome and to recognize them as foreign. And they probably come in through a process called introgression, where organisms mate with closely related species. And that results in the introduction of new genetic information into the genome. So the real question, there are two questions in your question. One is, well, how do you survive without a de novo? And the other question is really, perhaps you're implying, is that, why did this loss happen? Was there a purpose to it, or was it simply something that was tolerated?
Or also, if there is any counterpart that can balance for the loss.
Right. And so it is the case that Cryptococcus and related fungi also use RNAi to silence transposons, and they appear to be redundant. So it's definitely the case that there's parallel pathways that keep the transposons silenced. And that perhaps allowed things to be tolerated. It's also possible that introgression is rare for Cryptococcus for some reason, or if it happens, anybody who happens to be unlucky enough, an individual that's unlucky enough to acquire a new transposon simply dies off and is removed by purifying selection. Those are really hard. They're good questions, but they're very hard to answer. Whether there was any sort of advantage, disadvantage, or purpose to the loss. I tend to believe it's something that just was tolerated. And maybe it was tolerated in part because of the transposon content and the activity of the transposons that are there. Maybe they're not as dangerous as some other transposons might be, who knows? It's really difficult to say. And it's clearly a rare event. We haven't found another example of this in the fungal kingdom, although in certain insects, there's examples of loss of a de novo enzyme, and they only have DNMT1. And in some cases, people have looked and found that there isn't even any DNA methylation there. And in other cases, I'm not sure if people have studied it in detail. One big challenge of looking at DNA methylation is that the highly repetitive sequences are difficult to assay by sequencing, because the sequencing reads don't map uniquely always, even long reads. And so there's some technical challenges there.
Other questions?
Yeah, so one more thing. What urged you to think about that adding ATP will be helpful in its
yeah, well, it's because the enzyme has a switch SNF ATPase. And although we looked at it and we thought that's probably a chromatin remodeling function, it was a very good student. He tried ATP anyway, just in case it wasn't a chromatin remodeling function. And so I think this is a case where you have a switch SNF ATPase that is involved in coupling of events and not chromatin remodeling. We have no evidence that it binds DNA and that it actually has any chromatin remodeling activity. So you can't just look at a protein sequence and infer how it works. You actually have to get your hands dirty and do the hard work of testing, what might be going on.
Thank you, sir. That's all from my side.
So if there are other questions, or I should go ahead and ask 2 questions. So Hiten, I have a question. So I think you mentioned Cryptococcus has RNAi as well.
Yes.
So as in the yeast, RNAi feeds into Cly4 pathway.
It does not. In Pombe, RNAi is coupled to H3K9 methylation.
Yeah, sorry, in Pombe. Yeah.
In Pombe, yeah. But it's not in Cryptococcus at all. In fact, we've purified the complexes. They look completely different. And it appears to have nothing to do with chromatin and be a post-transcriptional pathway. And we don't know how it works, actually. We suspect it's controlling translation, but it's not coupled to DNA methylation or.
So in the RNAi mutants in Cryptococcus, you don't see any reactivation of the transposons.
Oh, you do. Yeah.
So it means DNA methylation or something is being affected there?
No, it just means that some transposons are better silenced by, so for example, there are transposons, there are DNA transposons outside the centromere that aren't methylated, and they're only silenced in a non-redundant fashion by RNAi. And then within the centromeres, you can detect an increase in transposition of transposons without RNAi. But as you asked before, we haven't done things like make the double mutants with an MT5 or the single mutant for that matter and compared, So I suspect it's going to be a quantitative effect and also an effect that is dependent on the specific transposon family, because I'm sure there's, the usual genetic conflict arms race going on between the RNAi machinery and the DNA methylation machinery and the transposons that they're meant to silence.
Yeah, it could be interesting to see which class of transposons are RNAi dependent, because for higher organisms, it may shed some new insight. So I have another question, which is more related to, meiotic inheritance. So if you explained us DNMT5 and then, passing the Cryptococcus through meiosis. I'm wondering if you did an experiment in which you had DNMT5 mutant cross to the wild type, and in the diploid, you have now a heterozygous state where one of the centromere is non-methylated and there is a wild type, which must be methylated. And you grow it, and then you take it through meiosis and see if the non-methylated copy, which was originally driven from the DNMT5 mutant, becomes methylated through meiosis as well.
So
did I make my question clear?
Yeah, I showed you that experiment. It probably went by quickly, but we actually did the cross and looked at such progeny, and they stay unmethylated. The way DNA meiosis works in mushrooms and our organism is that when mating happens, they form hyphae. So like in the soil, mushrooms form hyphae, and those hyphae are actually what are called dicaryons, where in the filament there's both nuclei from both parents in the same cell, actually. And they grow mitotically, and the nuclei are kept apart until the organism decides for some reason to make a fruiting body. And so Cryptococcus makes a microscopic fruiting body, but the mushroom that you eat is a fruiting body. And then it undergoes nuclear fusion and meiosis and forms spores. And so we put them through that process. And admittedly, we only looked at one region because that was what was convenient to do. We could have done more in terms of sequencing. And at least in that one region, in the 3 progeny we looked at, there was no acquisition of methylation. We should really revisit that and do some long read sequencing and methylation analysis to see if there's perhaps the equivalent of gene conversion that happens during meiosis, the non-reciprocal transfer of genetic information. It seems to me possible that there might be the non-reciprocal transfer of epigenetic information under that instance, and that just hasn't been done. I mean, it'd be a good experiment to do.
Because we have a similar phenomenon in Arabidopsis, where if you cross DNMT1 mutant, and you have successive generations where you lose DNA methylation, when you bring it back to the wild type and take it through meiosis, now the non-methylated chromosome will acquire methylation. That was something mind-blowing for me, that it's not only the chromosomal inheritance, but also the DNA methylation patterns are also being copied on non-methylated chromosome and then passed on.
But how much of that is actually like the chromosomes talking to each other versus the small? I wonder whether it's the small RNAs that are involved in RNA-dependent DNA methylation that are mediating the establishment in that case.
Yeah, the mechanism is not known. It was published by Hidetoshi Sase, I remember, 2003, Nature Genetics. I remember reading that, and looking at your data was, again, it came to my mind.
I'll have to look it up. I don't know that paper.
Yeah. Any other questions, ladies and gentlemen? And then if there are no more questions, so let me thank on behalf of all of us. Hiten, it was real pleasure to have you and to listen to exciting data. I really enjoyed looking at the structured data because you showed something. For the first time I saw, this flipping of cytosine and how methyl mark is being added.
Yeah, those are fun.
Yeah. So, and having read papers from Densha, that's amazing to see he's now doing this side as well. Thank you very much.
My pleasure. And again, thanks for having me. And sorry again, I was
No
late.
problem at all. Don't bother about that. Thank you very much. Good night.
Good night. Good morning.
Bye-bye. Yeah. Bye-bye.
Bye-bye.